There are over 800 species of reef-building corals, and coral reefs play such an incredible role in ecosystem. They support fish, mammals. They also provide incredible support to humans. They protect our shores, and they create the livelihood for over 1 billion people. And we're losing coral reefs at an alarming rate. Projections say by 2050, we may lose 90% of the coral reef from bleaching. Now, bleaching is not always 100% extinction, because if coral have a chance to rebound the next year with cooler waters and less acidification and no fishing trawling on them and everything else, if they don't have repeated insults, they can actually come back. And so we need to capture that biodiversity before it's really gone so that we can actually help bring those reefs back to fruition. And we're working right now with a project with Mary Hagedorn, a leading scientist at the Smithsonian. Let's hear from Mary.
I've been working on the cryopreservation of marine species since 1993, and I realized it was going to be important because you could tell the oceans were going to be in trouble from climate change. Initially, when I started, I was really just trying to work out the science of how we do this, but then it became very clear that things were changing very rapidly, and climate change and bleaching events were happening. And, I became very worried that we needed to get this science done quickly. I work on the conservation of coral, and I use human fertility techniques to do this. Basically, we are cryopreserving or freezing, however you want to think of it, gametes, the sperm and the larvae of coral, as well as small fragments of coral. And the reason we are interested in using cryopreservation is that it allows us to stop time, really. We can freeze coral and keep them for a day in our bank, or 10 years, or 100 years, or 1,000 years. So it gives us the ability to use this material today or save it for many, many years in the future to potentially reseed the ocean. One of the major outcomes of our process is scalability. We want to be able to cryopreserve coral easily and cheaply so that every professional that wants to around the world can form their own cryopreserved banks of coral and thus speed up the process of securing the biodiversity and genetic diversity of coral.
So Mary's project is one of 8 that Revive and Restore is funding right now. We call it the Advanced Coral Toolkit. And we have researchers working at understanding the underlying genetics that allow some reefs to be resilient and continue in the warming waters, some that may even show signs of resilience to disease. So as these scientists are working, they're developing the ability to create coral stem cells that could be used to repopulate reefs, as well as new techniques using proteomics and other parts of research to figure out how to combine these tools so that we can actually make a big difference